Thank you very much for the honor and the pleasure of the invitation. I'll try to take you through some of the thought processes that we need to be doing and some of the excitements that you heard earlier, based on the example of some of the pharmaceutical regulation. Basically, what I'm going to try and do is convince you that a lot of the things that we have heard are already I think going forward in different ways. However, the central aspect of actually storing, exchanging the data and looking at it from a central perspective is not something that at least has been built into our health system yet very successfully. So basically, I'm going to talk a little bit about innovative therapies, the potential and current challenges, the availability of new therapies in the EU, making them available to patients the way we do this in Germany with regards to the funding and finding a price for it. And then, of course, the big role that then the real-world data, if you so wish, has, and especially also some of the roles of patients and some of the needs of patients that we're currently not addressing. So innovative therapies with the potential and current challenges. The current treatment landscape is, of course, as you have heard, limited by some limits of effectiveness. It really neglects still to a large part individuality. Some of the guidelines that we're currently applying are really outdated compared to the current status of science, and therefore there is a delayed access across innovation, especially also if you look across the countries and if you look into different areas of countries, which also leads to a short-sighted focus. At least in Germany, we oftentimes have this initial cost discussion that neglects the long-term benefits of what we're trying to do. The limited treatment options for rare diseases and the funding options for such treatments, eventually we have to keep in mind leads to a human cost because we're not implementing as fast and as well as we could have. The potential of advanced therapies is, of course, as you've heard earlier, innovative medical innovations and interventions from the diagnostic through the therapeutic offer personalized and non-personalized approaches that will be able to do many more things that we're currently still only dreaming of. And that has, of course, to do with targeting molecules, targeting immune structures, targeting all kinds of functionalities, with the goal in mind to improve patient outcomes. That is in cancer, for example, higher remission rates. We want to reduce side effects. We cannot necessarily always obtain a cure, but of course that is potentially very important to still perceive and aim for that goal. Reduction in overall healthcare cost due to improved treatment efficacy is something that we've actually seen for a long time and has in part compensated for the rising need for healthcare by, for example, allowing us to do many things today in the outpatient setting that would require inpatient treatment earlier. And so we're really already in the middle of this process. Unlocking the precision is, of course, the role of genomic diagnostics for personalized medicine, and you've heard in the previous talk, so I'm not even trying to repeat or copy that because it was so brilliant. We're trying to unlock the individuality and make smarter treatment decisions. We're trying to predict risk, although, as you have heard, that is something that has originally been overestimated, and we're still quite not there yet. We need to find, especially important, the right patient for the right therapy, and make sure that the personalized therapies match the right person and the right person can be found. And we, of course, want in this whole process to drive innovation from real-world data, from genotyping informations to next-generation therapies. In cancer, for example, we have traditional cancer treatments that have broad categories of disease, limited ability to account for genetic variations, and of course leads in most often to overtreatment or undertreatment. We have seen a very effective introduction of personalized medicine in oncology with genetic testing now in many diseases, with selection based on not only the genomics of the person, but actually the genomics of the disease at the actual stage, with more precise dosing and, of course, targeted treatments that are trying to hit the right structures within the cancer. And we have seen something in Germany, and I know, of course, of many other networks across Europe. In cancer, we're also seeing this in other diseases where academic networks of precision medicine are currently monitoring, guiding, and then sometimes even performing the treatment for the majority of patients in a particular disease. So you have this interesting situation that non-government organizations, sometimes even patient organizations, fund and organize the science around the treatment that is affecting the majority of patients. This is, for example, the German Lung Cancer Network that has basically negotiated a contract with the health insurance companies to make sure that all patients across Germany with metastasized lung cancer not only get genetic information derived from their tumors, but that there's actually a central structure that quality controls and looks at the content of that data and advises the regional hospitals and physicians of the capacities and potential of such treatments, and making sure that wherever that's possible, these patients with particular findings are being enrolled into the clinical trials that offers them the access to this particular treatment. And so I think we're also coming to a moment where the availability of new therapies in the EU from the pharmaceutical market is basically something that health systems need to grapple with. On the pharmaceutical side of things, the expenditure, we're always discussing this as if pharmaceutical drugs are breaking the bank and killing the system. Across Europe, the number is between around 12 to 13%, and in some systems up to 25 to 30% of expenditures are spent on medication. And then a little bit more, so roughly less than a quarter to a third of the overall expenditure goes into diagnostics and treatments of this thing. And 3/4 of the expenditures that we have are actually going into such things as hospitals, staff, and other issues. We have discussed earlier, you heard the discussion on what is the cost to conduct a human genome. I can give you the numbers for Germany. You're talking about billions of billions, but if you look at it from a citizen perspective, we are spending roughly 5,000 euros per year per citizen on healthcare, and we're spending about 1,000 euros or less on drugs and diagnostics, just to give you a number per year, to give you that understanding. We have an interesting mechanism in Germany that once there is market access to a drug, the company can basically set the price for the first year on their own. So therefore, at that price point, we have very quick market access in many of the European countries. And so there are mechanisms we know, not on the diagnostic side yet, where this isn't working as well, but we know that we can get commercial products to market if we design the rules right. So this is like, 47 days, I think, on average in Germany. And then, of course, there is the process of health technology assessment. We call it AMNOG in Germany, where data is being used and can be submitted by the company to set a price point. The primary goals of the system is that there is cost effectiveness across the system and making sure that patients can actually get and afford the drug, and we can maintain a high quality healthcare across the system and control the expenditure. The idea is that a manufacturer can only command a higher price if the treatment is better than the previous version of the treatment available for this particular particular disease. The issue about that is that usually that process involves data from phase 2 and 3 studies in order to show performance. And of course, that is something that is very hard to come by in diagnostic settings, in settings for rare diseases, and in other types of situations. And therefore, this benefit assessment and technology assessment, I think, needs to be approaching new grounds with regards to looking at real-world data, looking at outcome data from across the health system, and so forth. The impact for patients of such organization that makes sure things are getting applied across the system early and then setting a fair price for it is, of course, that they get access to this situation. And we just heard it about the sequencing, and it has a number of impacts for healthcare providers. The problem with these sort of model of getting pharmaceutical drugs to market is it works in a regional system. As long as you can put it in the pharmacy and the doctor or the hospital can order it, it works, but you don't need a central system to look at the data, to have a database to compare things. And so therefore, I think a lot of the things that we're talking about today, personalized medicine, using data in the right way, is something that is not yet reflected in that system. And we need to innovate, and we need to build structures that allow to do these processes, as you have heard. And as you have heard, some of these pilot projects or academically driven projects are happening now across Europe, but I think all the local health systems are still grappling with the reality of putting a more central structured system into place that then works across Europe as well. So the power of real-world data is what I'm trying to get at, is the disadvantages of randomized clinical trials, as I told you, in some settings are not possible because of a limited patient population, because of the artificial structure of the settings, because of the limited availability of long-term data that is collected just on purpose and not sort of generated from the system automatically. And of course, in rare diseases, it's not possible, or in some instances where the new treatment is especially effective, is also unethical to conduct. The bridging the gap is, of course, that real-world data, if it has sufficiently enough quality, can supply some of that information, and in some instances, depending on the statistical methodology, can actually help us to unlock the full potential with regards to that. There is a lot of research going into causal inference and other types of methods. If you follow the current literature, you can see that the artificial intelligence models, even some of the newer implementations of large language models, are actually very much honed into identifying differences in patterns between different sets of data. And so therefore, we are, of course, hoping and expecting that some of this information will also blend into the system at this point. So we need to build trust through transparency, because these are, of course, large data processes, and the research advancements will be a strong selling point in establishing such things. And I think there's one thing that we are completely overlooking, which also plays into the question of getting a diagnosis for people with rare diseases, and that is the right to be found. We have always argued data safety in terms of the right for the citizen to refuse to share their data in order to be safe. People with cancers or rare diseases or life-threatening diseases are not safer when they do not share their data. In fact, sharing their data is, in some of these instances, their only chance for survival. But we are very bad of actually making sure that even if they offer their data, or their data is in the health system, that they are actually found. So there is, of course, the group of patients where we know they have a rare disease, but there is an almost as large group we can anticipate and expect where the patient isn't even aware that the current disease that they have is actually part of a rare disease. And so in that sense, their data should be scrutinized and they should have a chance to actually be found, to be able to participate in studies and basically have access to all that the health system has to offer, which in many cases, as you have heard, in such diseases and such data processes is still on the academic and research side. So, in conclusion, the advanced therapies, the key to progress is, of course, that we get enhanced patient care and a more personalized medicine and patient empowerment in this field. I think we also have to make sure that prevention and catching the cases early becomes a part of our routine in the health system. We need to rethink the health technology assessment and regulatory frameworks towards faster approvals. We've done that reasonably well in many of the health systems across Europe in pharmaceuticals. We have to also get an understanding of the long-term benefits and of personalized medicine to make sure that we're really reaping that process and seizing the opportunities. There needs to be collaboration. There is a chance, of course, to do it across Europe and even across the globe for the more rare diseases and of course also for what the companies do. There is the vast potential that we're not really tapping into at all right now with regards to the real-world data that continues to be collected day in, day out through the health system. And also, I think part of the key to that situation is that we need to develop standardized protocols to try and understand how can we put the data together, where are these patients, and what can we do for them in an ideal scenario. Thank you very much for your attention.
After hearing so many breathtaking talks, I feel a bit humbled to be speaking about the topic of health data. And I'm going to speak mostly about the European health data space. It's come up many times already in our discussion. And I would like to share with you what it's about. So in a way, I'm describing the regulation. But I will put a few personal remarks in as my reflections on it. The first thing to say about it is that it is actually gripping many people I engage with a lot of excitement, and that includes me. So even if I say a few negative things occasionally, my overall feeling is this is a really exciting breakthrough for Europe. And I just want you to be minded of three key objectives that it has to give people much more autonomy and control and access to their own data, to provide a consistent framework for how we all use health data for a variety of purposes, and to also stimulate innovation at a coherent European level. So let's keep those three things in mind. I'm going to take you on a story that also breaks down the EHDS into its two key parts. It has a part that focuses specifically on identifiable data that travels around Europe to support the care to the individual, and data sets that are accessible, using Tim's word, around Europe for the purposes of learning from the data. Two very different, it's a single document, but really two very separate parts, and it's useful to remember that. But the first thing I wanted to highlight to you is a couple of definitions. The definition of personal electronic health data, a new term to me, despite 30 years in the field, electronic health data, but okay, I'll go with that. There is explicit mention of genetic data, but nowhere in the regulation is there any distinction made between the single assays that are looking for particular genetic biomarkers and whole genome sequencing. So just keep that in mind, that it fails to differentiate the two from a data protection perspective, from a patient utility, from a research utility perspective. And secondly, it draws a distinction between the primary use of data, meaning data for the provision of healthcare, and secondary use of data, meaning data to be used for other purposes. And so the stuff in red sits in a slight no man's land between those two, not explicitly catered for in the regulation as such. So let's turn first to the patient-specific thing, that individuals have more control over their data and there is better exchange of data to support healthcare delivery. Electronic health record systems, which are the birthplace for a lot of the clinical data, must now be certified, well, not now, but in a few years, certified against two key requirements. One is the ability to import and export a priority set of electronic health record categories of data, which will be embedded within something called a European Electronic Health Record Exchange format, which will be upgraded soon. And secondly, the ability to provide transparent access logs to patients. Who's been looking at my data? There are also some, I would say a little limited, but some data quality provisions that EHR systems will need to be proactive about the data that they're capturing. Other kinds of technology, if they talk to EHR systems, also have to obey the same rules. So that is also a bit of a shake-up to the medical device sector. It's a bit of a shake-up to AI, that it has to play an interoperable game with EHR systems. People have a number of rights. They should be allowed to access their own data in a consolidated form. That's going to push member states a bit, because up to now, people can access their data on paper or PDFs or other ugly formats from every single place they've had care, nothing consolidated, nothing computable, and there are extended GDPR rights. And also there is now in the final version of the regulation, the April copy, a requirement for member states and for the Commission to actually invest in training people so that they understand data and digital better than they do today. Absolutely vital. A couple of drill downs. One is, what is the data that is likely to be in the exchange format? There already is an exchange format, and there already is a European patient summary, but it's now 10, 11 years old. We're all expecting that the revised secondary legislation will adopt instead the ISO patient summary, and you can see here, maybe not very legibly at the back, a bunch of categories of data that it contains, but it's pretty standard medical summary stuff, plus some of these clinical documents that you can see, but the computability of those is not necessarily to be enforced. So you should be asking yourself, is this going to be a real asset to the kinds of innovation you've been hearing about this morning? But also, let's turn to the topic of patient rights. It's very relevant to the interactions of the last few minutes. Patients will have a right to place restrictions on the access of their EHR data, and that can include restrictions to certain categories of data, e.g. genetic data, which is singled out in the regulation only as a throwaway example. But of course, it's not a throwaway example if it's explicitly mentioned. It's like saying, as a patient, despite being happy to share your clinical data, you might be nervous about sharing your genetic data. You can say no if you like, which of course guarantees that everybody feels nervous about sharing their genetic data if you highlight it in that way. So we need to watch out what the signals are to society here. But there's also some good news, because people will have extended GDPR rights. They will be able to download this consolidated computable data in the exchange format representation themselves. So they will have their own data and they will have autonomy. That means they could be found. They could then nominate that I want to offer my data to another party who's using it for research, or to a data altruism organization defined in the Data Governance Act that will hold my data on my behalf and make it available to others. So it also opens up a new channel for data access that might be useful. Let's jump to the secondary use. I mean, it's really hard to summarize nearly 100 pages of regulation in several minutes, so I'm doing my best, I promise you. So this is about how can a diversity of actors, from research to policymakers, public health, etc., be able to analyze data at scale, at a European scale. So the first provision that's really important to highlight to you is actually in bold. It's an obligation. Anybody that holds data of certain categories, which I think is my next slide, is obliged. The word obliged or obligation is used in the regulation to make that data available for secondary use. That is going to cause, and it is causing, some anxiety among data holders already, saying, but hold on a minute, I'm still in the middle of using my data, or my data contains trade secrets and so on. And there are provisions for that, but they're entirely imprecise. Data sets must be labeled with fair metadata, etc., and will be put in a catalog, but data quality labeling is optional, which I find disappointing, because it means you don't have any assurance about the quality of the data unless bottom up, so not top down in the reg, but bottom up as a culture, as a community, we decide we will holistically quality label our data sets. That is a bottom-up possibility if we choose to do it. The channel for data access will be through national, can be regional, health data access bodies. They will hold the catalog, people will make requests through a health data access body, and they will then provide the data back, which can be anonymized, it could be pseudonymized in a secure processing environment. And they have to provide some transparency to the public about requests, refusals, and what's happened to the data. There are lots of categories of data. I'm going to flick through these really quickly. What you need to understand here is that it's pretty much every kind of data in a vaguely health space or health impacting space is somewhere on the blue. And almost all of the places where the data might live are somewhere in the middle box. So it's pretty comprehensive in its coverage of what's in scope. There are permitted and prohibited purposes to safeguard the public. There are a bunch of permitted purposes which are relatively straightforward and logical, and research is in there, don't panic. And the prohibited purposes are also reasonably straightforward. They're the kind of things that could cause harm to people or population groups, and they are prohibited. What I'm hoping is that these kinds of lists will inspire public confidence. Why do we need to inspire public confidence? Because people can opt out of secondary use of their data. And that could include picking industry and saying, industry can't have my data. Well, we've already heard today that industry is quite useful in our field, haven't we? So that would be a strange and disappointing choice. But also, they may opt out of certain categories of data, like genetic data. So you need to then consider, how do we persuade people that they shouldn't exercise that right? That's not a top-down issue. That's a bottom-up issue. How do we inspire confidence? How do we communicate that data can save lives? Please don't opt out of your data being used. I want to jump to a second topic fairly quickly. I appreciate that I'm running close to end of time, but I still have a few minutes. The AI Act is also a fresh kid on the block for us. AI is very exciting, and I'm sorry to say I'm not going to speak about the AI excitement. There are lots of exciting speakers today. I'm telling you the rules. Pretty much every use case of AI that I've come across in health or projected is what we would consider high risk under the Act. That means you've got to undertake a pretty rigorous process. But one of the things I'm very concerned about, and with colleagues, is how can we inspire patient, clinician, public confidence in using AI in a health setting. And I believe that clear information is a critical part of that. So we are working in a project to look at that topic. What might be the content of the clear information that would convince people, convince a patient to use a device or an app that is maybe even closed loop, or have a patient confidently sitting with their clinician knowing that AI is somewhere inside their decision-making process. Clear statement from the developer, what is the objective of this AI? What is it good for, so you know when to use it and when not to use it? What kind of patient have I developed and tested this on? Because, you know, if it's been developed for adults, don't try and use it in kids. If it's been developed for mild disease, don't use it for severe disease, and vice versa. That stuff is very important. It needs to be, we need to have a method of defining what that third bullet means. Just saying, yeah, trust me, I used good quality unbiased data isn't good enough. How do we measure that? How does a developer prove that? Would be an interesting one. What is the role of this algorithm in the position of human decision making? Is it influencing? Is it advising? Is it taking control? People need to know that. Where can I go to find out how you've experienced the success of this, how it's been effective, how it's been safe? Who's approved it? Have you been through regulatory and HTA or other approvals? Have professional bodies endorsed it, for example? And is it clear to the deploying healthcare environment, how do you wire this up? How do you plug it in so it gets fed the right data and therefore is most likely to perform well? And are there driving instructions to the clinician or the patient? So it's really clear how to use it, when to use it, and when maybe to be not so clear about using it and to say, I'm going to override it here. And finally, where does accountability lie? We want to see if we can formalize those things in order to help in the future. Now, I want to give you a final slide. Well, actually, two if I have time. I'm running out of time. One is about where I think this plugs into this conference. So we've heard a lot at the biological markers and so on. We've got to consider that in order to interpret those, we need clinical data too. And you call it phenotypic data in some of your circles, but it's routine EHR data, warts and all, as we heard. And increasingly, we're going to get data from people as well. And that all needs to come together in a coherent environment. That means we need to be able to do things like federated networks and big data analytics, of course, not moving the data, but running queries on the data, and then making good use of that data for a diversity of purposes. And what I'm hoping is that this is the landscape that EHDS will help to power. So that's really my hope. But you need to answer the question, have you heard from me stuff that gives you hope of this? Or are you feeling the regulation is going to be a bit limited and clipped compared to what I really hoped? My final slide is really to be left up for discussion, so it's good that I'm now running out of time, or it's up, it tells me, which is really, the EHDS is not alone. Some of you in this room are responsible for some of these other logos. There are other logos that I couldn't fit on the slide. And so I want to stop talking and say these might be the questions you ask me, but they don't have to be the questions you ask me. Thanks very much.
I hope today to enlighten you a little bit about our efforts in the European Union about generating 1 million genomes. It was mentioned several times already. I'm speaking here on behalf of my colleagues in that. They're all sitting on the second row in the middle, so you can approach them afterwards. Of course, what you see here is what we've been talking about today already. We are searching for evidence and events with usually very immature tools. Then we find something, we try to decipher it, and then in the end, as we heard already today several times, we're always confronted with different sides of the coin. There's always a Hippocrates and a Pandora side to things. A few disclosures, I sometimes get paid for getting to meetings from companies and to also give them advice, but I get one salary from Erasmus MC and I hold no stock whatsoever. This is of course a starting point. It was referred to today already several times, the fantastic human diversity we encounter every day. Look at your neighbor. You see here some fine specimens of that, and of course they are not only different in the way they look, but also in the way they behave. But of course, importantly also for today, the types of diseases that they will get. And what we've learned, to summarize, let's say, a few decades of genetic research, is that DNA variation is underlying in a causative way many of these aspects, not only in development from birth onwards, but also in the appearance, and that information is currently used by forensic science, for example, in a totally different corner of society. Their behavior also is under genetic control. That means it's to some extent determined by the variations in your DNA. And then aging and diseases is, of course, the most relevant theme for today. And we've learned through many efforts, and I'll show you a few of them, that DNA variation is indeed in a causative way related to the risk of running such diseases. All of this has led to this concept of, it was mentioned already, a precision medicine. I don't like the term personalized medicine, or maybe even better, genomic medicine, and that's what we're mostly discussing today. We're approaching lunch, so I'd like to enlighten you with how we eat lunch in Holland. That's usually with a sandwich with cheese and some milk, and that's all fine because that leads to a very high calcium intake, more than 1 gram per day. And if you travel a little bit south to here in Belgium, you notice that lunch behavior is a little bit different. And that's all fine, of course, but the result of that is that the dietary calcium intake in Belgium is about half of what it is in the Netherlands. I show this slide because it highlights the importance of environmental factors, and sometimes overlooked. We can now sequence a genome, it was mentioned already, in 24 hours for a very reasonable price and a very high accuracy also. But getting environmental data is really challenging. I forgot how many beers I had that evening, and I did not remember what I ate after having those beers. But that's the way how we collect environmental data at the moment. So that's why there's an enormous emphasis on genetic data and also many efforts, but very little on environmental data. Not saying that it's less important, but it's much more challenging. However, maybe also here genomics can start helping us because there's now microbiome sequencing, and that might help us to understand what is the dietary composition by analyzing stool microbiome. We can look for virus particles to see where those particles or those dietary ingredients have come from. So also here genomics might help us to improve that field of environmental science. Coming back to what also was mentioned briefly, that classical distinction between monogenetic diseases or simple genetic diseases and what I'm mostly working on, complex genetic diseases. You see here a few characteristics listed which make them comparable, but of course not similar. You see here that what we've learned from DNA analysis, that for these rare diseases, the Mendelian disease, there's usually a causative mutation with a very severe effect, but very rare, luckily. But it travels in families, and that has been very instrumental in identifying them. On the opposite side, we see that those complex diseases have a genetic component, meaning that they're not completely determined by genetic variation, but there's interaction with genes and environment, and there's not one DNA variation involved, but there are hundreds, if not thousands of them. Of course, we've also learned that things are not so black and white as I depict them here. Also, Mendelian diseases are influenced by the rest of the genome, for example, in determining the penetrance of that mutation in that particular family. And on the other hand, those complex diseases also have some more rare characteristics driven by, let's say, rare genetic variants of a slightly bigger effect than what we normally see with the polymorphisms. So, of course, polymorphisms and rare mutations are together making up, let's say, the genetic architecture of diseases. We've identified a few tools in the past decades that have allowed us to identify the DNA causes of those diseases with very high accuracy now. It was referred to already by the previous speaker. We have these Mendelian mutations that travel in families, and by sequencing we can identify them very rapidly now. If you have all the DNA of the family, usually within a few days or a week or so. On the opposite side, you have here the common complex diseases have been subjected to what we call genome-wide association studies, so taking a much more holistic view and let the genome tell you which variations are involved or associated with that disease. And because it is DNA variation associated with a phenotype, we know that it is a causal association as opposed to classical epidemiology. So GWAS has been very instrumental in overloading us with all these genetic variants, and they have led to this concept of polygenetic risk scores, where we combine all those genetic variants into a score for a particular phenotype or disease. And I'll come back to that at the end of my talk. So this leads altogether to what we call genetic architecture of any phenotype or disease. So here on the x-axis you see, let's say, the distribution in the population going from, let's say, a phenotype with a low value, let's take low cholesterol level, to very high cholesterol levels. And then, of course, in the middle you have the vast majority of the population having an average cholesterol level. We now know through all kinds of genetic studies that at the end of those distributions, we have these typical rare Mendelian disorders leading to these disorders, very rare. And in the middle, let's say, your average cholesterol level will be determined by hundreds of DNA variations in your DNA, which we can now determine with a polygenetic risk score. Now, those polygenetic risk scores have been developed for, you see it on the left, for not only risk factors like cholesterol level or bone density or what have you, but also for diseases, disease phenotypes, and that has led to this abundance of these PRSs now in databases which we can start using for various purposes. At the bottom you see the technology that is used. I will not go into that today, but suffice to say that polygenetic risk scores currently are really the hot item in genetics. Now, this is what we eat for breakfast in genetics land. So we're looking at these DNA variations and looking them in genome sequences, usually whole genome sequences, sometimes exome sequences. And what we've learned is summarized here in this table that we've sequenced only very little humans at the moment. It's about 5-6 million maybe. What was it last week? Regeneron released another data set, so maybe there's another 500,000 added to that, but it's in the few millions. Opposed to 8 billion people living on the planet. So our understanding of DNA variation between humans is very limited at the moment. But already on that very thin basis, we see that there are 350 million positions in the genome where DNA variation can occur. So this saying that our DNA is 99% identical is total bullshit, in fact. We differ a lot. There's much more DNA variation than we currently know because of our very limited efforts in generating those DNA sequences. Let's go back to the 1 Million Genomes Project, but before I'll go into that, a map of Europe showing what we call the genetic density per country, so per capita. We are looking into, this is an active process, by the way, a work in progress, together with Nicole Soranzo from Human Technopol in Milan, looking at how much data is there, in fact, gathered across the European continent, including England, I must say. And you see here in the brighter colors, green colors and yellow colors, there are some, let's say, enlightened examples, but they are exceptions. Of course, the UK is a big one in there, and I'm very happy to see Tim here, because you're a shining example of what we should do at the European continent, actually. You also see Iceland there, and there's Estonia there, where there's a very high density of genetic data because of their biobank led by Andres. But you also see that there's a lot of, let's say, depreciation of genetic data across other European countries. So in other words, we need to get our act together and start sequencing and generate data for that. And that is, of course, a major driving force in the setting up of this 1 Million Genomes initiative that was launched in 2018 by a few signatory countries, and that group has been expanding now to include all European member states. And in that European initiative, we have identified a few working groups which are working on different aspects of those generation of the data, of storing the data, and for what kind of use cases might that data be useful. I will not discuss all of them. I will focus on one in particular today. That's the Genome of Europe, which was launched a few years ago, in which we decided, well, let's start with making reference genomes to capture the European diversity. And why would we do that? Because, well, actually, looking from a genetic perspective, there's a lack of insight in genetic diversity of the European continent. We have, of course, some databases with that, but they're mostly American, UK-based. There are European samples in there, but there are not many, and they certainly do not capture all the diversity across the continent. So that was a big driving force in starting this project. It also is important that we decided that it should be representative of the European diversity, whatever definition you might take for that, and it should also be proportional. So let's say Germany is expected to deliver more genomes than Malta. We also wanted to know what kind of subgroups of populations are living across the European continent, because, as you all know, we have a very rich colonial history in the continent, and of course there are very recent migratory streams in and out of the European continent. So we also wanted to capture that because they are very important consumers of healthcare, of course. So with those, let's say, criteria to start such a project, we also thought of how to organize it and what kind of use can be made of that. Well, a very simple use is simply looking up DNA variations. You do a sequencing of a family somewhere, you find all these thousands of variations, you do a quick look in the European reference database, and then you spot a few of those variations which have been seen before in normal, healthy citizens. So that leads you to focus more on other likely pathogenic variants. We can do, of course, also basic population genetics with it and define population subgroups based on that. Very important for healthcare, I think, is the recalibration of all those polygenic risk scores I was referring to, because they are coming out of GWASs, and we know GWASs are quite biased in the composition of the source population in being European Caucasian, mainly from European biobanks, but also American and other biobanks in there. They're not really multi-ethnic and certainly not representative of the variation on the continent. And last but not least, we also want to use this reference database to start imputation panels. And you can approach me afterwards to explain you why we are wanting to do that. A few basic characteristics of the database that we want to generate with this. Well, first of all, it should be a normal control population. So it's not a collection of cases, but it should be control populations coming, for example, from population-based cohort studies. And Europe is indeed blessed with many of them on the continent, but also elsewhere, of course. And you see a real surge now in a lot of biobanks being generated and started. And again, UK with UK Biobank is a very good example of that. But you see them elsewhere also. It is in fact important for all the use cases that we previously defined in the 1 million genomes project. So for rare diseases it's important, but also for oncology, but of course also for complex diseases, for example, for recalibrating those PRSs. The initial focus of the reference database is not so much on phenotypes, but is on getting genetic data together with limited phenotypes, age, sex, and country of origin. And later on, of course, we ask the people to contribute to choose their samples wisely, so to take them from population cohorts and biobanks which have a very rich collection of phenotypes. So later down the line, we can start connecting those. We will be working with various sequencing technologies, short read and long read for the aficionados. The exact proportion of that has not been determined yet. We just were awarded a grant, so we're discussing that intensely. We were initially aiming for 5%, but this is probably going up to 10% or maybe even 20% in the total database at the moment. And you see the 4 companies there which we are discussing this with. We're going for a clinical grade genome, so a good coverage, so good quality data so that they can be used for clinical purposes also. And of course, we also allow different sequencing centers across different European countries to contribute to that, which is a challenge, of course, but I think it will be closer to reality somewhere down the line when all national genome programs will be initiated. Complicated table, but I do like to show it to you because it was the result of a lot of hard work by first asking to these countries, how does your population look like? Which subgroups of population do you have there? Usually coming from the Central Bureau of Statistics. So in the columns here, you see the contributing countries, which are in the 1 million genomes initiative. And on the rows, you see the top 40 of all the different subpopulations they identified. So of course, in the Netherlands, we have Dutch people, but you might also see we also have Moroccan people and we have Turkish people living there. And that holds true for several different European countries, of course. So what this leads to is that we ask these countries to contribute those subgroups also to the reference database. And for example, this also highlights why we do this on a European scale, because Denmark, Germany, Holland and other countries can all contribute Turkish samples, although Turkey itself is not a member of the European Union yet and not contributing to the 1 million genomes initiative yet. So this interesting aspect of this reference database is really of added value for capturing the diversity. You see a lot of other subgroups there, which also come from the African continent, and again, pointing to this colonial history we have there. So this together leads to, of course, a very nice overview of genetic diversity of people that are living on the European continent. You see two red crosses for obvious reasons. One is China. We have some hesitation of collaborating with them, but we certainly will do this somewhere down the line. And of course, the United Kingdom, because of this stupid Brexit. But we're working on that. So this leads to this European project being awarded recently, the Genome of Europe, and we were funded to generate the first 100,000 genomes of this project. And the nice thing is that this is a matching program, so we're not only getting money from the European Union, but we also ask those countries to match the investment in there, and that works very nicely. So the total budget is 45 million of the project. We have 29 countries involved, 26 member states and 3 non-member states in there. We have designed a few working packages, of course, trying to deal with all the different aspects of generating those data. I'll not go into the detail. And we're currently in an administrative phase negotiating with the European Commission exactly how this budget will be looking like and how the whole administration will be dealt with. And we hope to start generating genomes shortly after this summer. A few acronyms you might have stumbled into while looking into the 1 Million Genomes initiative, because we have so-called coordination actions. And the first one is the 1 million genomes, one following that is the beyond 1 million genomes. And we have just a recent one being submitted by Serena, she's there, and colleagues, of course, to again get a coordination action funded. And those 2 projects, the GDI, the Genomics Data Infrastructure Project, it was mentioned already, and the Genome of Europe are 2 funded projects with a start and an end date, which are part of this initiative, just to highlight that to you. Of course, capturing diversity is a challenge. You see here a map of Europe called Eurotopia. I'd like to show it to you because it is one way of reflecting the diversity we have. And this map is made by Freddy Heineken. I think you might know him from the beer. And together with a few historians, he redesigned this European map based on, let's say, cultural differences, language differences, and other information. And that's, of course, showing already that we have quite some diversity in the European continent. Already this effort identified 75 member states, so to speak, according to these characteristics. Of course, this is reality, what we're working with. This is Europe based on prejudice. Meaning, for example, we in Rotterdam do things much better than they do in Amsterdam, in the Netherlands. And of course, it's a very used fact of life. And that's also happening in Europe, of course. This map is made by a Bulgarian artist, and I really advise you to look at the website. It's quite hilarious. On top of that, of course, politics is not really helping us. We had Brexit, I mentioned that already. So suddenly we cannot easily collaborate with our dear English colleagues anymore. There are walls being erected. Sometimes subpopulations want to go separate in the European Union, complicating things. And that is, of course, also countries that really want to join the European Union, but are not there yet. So it is challenging to start collaborating there, but of course we are reaching out to those people outside of the European Union. So we are, of course, having close friends with people from UK Biobank and Genomics England, and also was happy enough to meet Mr. Obama, who started this precision medicine program, and I also discussed the map of Europe with him and the All of Us program, of course. So we also hope to share data with each other somewhere down the line. So yes, we are seeking collaborations even with countries outside of Europe, and also Turkey is interesting to mention here, because once word got out that we were starting this project, they immediately contacted us and wanting to collaborate with them. So this is really fantastic, and that oil stain is expanding at the moment, I can tell you. A final few words on the polygenic risk scores. You see here what they are. So you see here people, individuals reflected on a axis of a PRS score. So on the right you see people with a high risk score. So these people have, let's say the top 1 or 2% or so, have a genetic risk which is equal to that of a Mendelian mutation. This is sometimes underappreciated. But then it's happening in many more individuals in the population. So it might be worthwhile to inform those people early on about this very high genetic risk. On the other end of the spectrum, of course, you have people with a very low genetic risk. So, for example, speaking about population screening programs, it might not be useful to include those people in the screening programs because, for example, mammography and breast cancer might for those people not be of added value in there. So these thoughts are now being elaborated on in applying PRS information very early on in a person's life to see if that can be coupled to, for example, population screening programs in order to make that much more targeted. A recent paper identified, let's say, the top 10 of these PRSs where most data has been identified and also diversity has been included. But that top list will certainly grow in the coming time. So what is the important driving force, it was mentioned already, is not just sequencing. We will do sequencing, of course, massively for the Genome of Europe and for other big projects, but arrays are sometimes underappreciated in this realm. I would mentioning this number of a few million people having been whole genome sequenced, people having an array data set runs into over the 100 million. So more than 100 million on the globe currently, like you, the moderator, have a genetic test. This is based on array data. That array data is for more than half in commercial hands. So sometimes we collaborate with them and we get their data, but sometimes not. But this is a very rich data source, which was more than instrumental in the GWAS era. So this price going down of these arrays, and our group in collaboration with some other groups were instrumental in it, in negotiating this with these providers, has been really a driving force in bringing data to the market, so to speak. Not only the academic market, because of this price level of less than 30 euros per DNA sample, this also becomes interesting in, let's say, more application areas across healthcare. So there are a few advantages of arrays over sequencing, but I'll save it for lunch to discuss with interested people. I'd like to show you here what we are thinking currently of applying genetic information in healthcare and especially prevention. You see here on top what we are currently working on in different hospitals around the globe, I can tell, in Europe, but also in the States and other countries, is using this information in patient healthcare. So, for example, applying a PRS for breast cancer in combination with a sequencing of breast cancer genes to see, can we re-stratify those women who carry a mutation like CHEK2, for example, which is not so penetrant. And this is piloted currently at the clinical genetics department in Erasmus and also in other countries I know. So you see applications where those complex genetics fields and the monogenetic fields are meeting each other in clinical diagnostics to improve diagnostics. Pharmacogenetics is an obvious area of application, of course, blood group typing and what have you. I think especially important are the applications in the screening programs. Several European countries and other countries have screening programs, mammography for breast cancer, stool tests for colon cancer. This is where personally I see the biggest impact of using genetic information, probably generated with arrays rather than sequencing, to have an impact on prevention of disease and lowering disease yield in the population. And of course, I mentioned briefly already, there are areas outside of clinical science, like forensic science, where there's a big interest in using this data also, for good purposes also. But we can have big discussions about that later. Finally, we're all using this combination at Erasmus, very local, in our own hospital here, and we are running some pilot studies to see what kind of information and what kind of clinical trajectories in the hospital can be improved by using genetic information. The idea there is that before the patient is going into the hospital, first a genetic profile is being generated, so a genome-first approach, and that information is then curated, of course, and interpreted by experts in the hospital in combination with databases and other experts. And that information then in a very easy digestible form, not taking more than a minute to digest and explain maybe, both to the physician and the patient. And of course the diagnostics and the treatment can be improved of that, but maybe also some lifestyle advice can be given to this patient because of secondary findings we do. Somebody comes in with a cancer diagnosis, but is also seen carrying a very high cholesterol level due to cholesterol FH mutations. So all this is piloted, and we have a few trials already running in the hospital to see how this all pans out, because there are many hurdles that was discussed already, which is totally outside of science, but how do you communicate? What kind of legal hurdles are there to do this actually? But there's a lot of progress in this area, and this is not only happening in our hospital, but really worldwide a very big movement. And this is also highlighting why we are doing this 1 million genomes project, to also get national health systems used to working with this kind of data. So with it, I would like to leave you not without thanking, of course, my collaborators. I'm always standing on shoulders here, and I'm very much open to any question you might have. Thank you very much.